what we have talked since the beginning. So we have the DNA, including genes, and then the part of the DNA which has the necessary gene will be transcribed to messenger RNA, and then messenger RNA will move to cytoplasm to be translated and to produce the polypeptide as a final product. If you want to have an imagination of the number of the nucleotides of mRNA, is like something approximately 2,200 nucleotides, which will be translated into 730 amino acids. If you remember, each 3 nucleotides is responsible for 1 amino acid, so that's why you have to just divide 2,200 by 3. So you have the length of the 730 amino acids per polypeptide. Another important thing is not that all the genes will be activated all the time. So only certain genes in the genome need to be expressed according to the necessities or according to the cellular specialization and based on the environment and whatever cell needs. Then the related gene will be activated and the mRNA will come and transcribe and then finalize with the protein product. For example, if you look at the pancreas, the cells inside the pancreas, they are responsible for secreting insulin. So not that all the cells are responsible for secretion of the insulin. So that's why only pancreas cells will have been specialized, and the mRNA will be transcribed from those cells in order to produce a protein which is called insulin. And because it needs a lot of insulin, so there will be lots of copies from the mRNA in order to make more and more amount of the protein. I think we have already discussed on this slide. We have 64 different codons, and it can be shown in a table such as this. And each 3 nucleotides will carry and responsible for 1 codon. And that codon will be responsible for carrying an amino acid. The number of the amino acids is almost 20 amino acids, and we have a combination of the 64 different codons. So why do we have 64 different codons? Pause the video and answer this question. Yeah, you got it right. We have 3 place as a codon for amino acid includes 3 places, and each place can have any 4 different types of the nitrogen spaces. So each place, it could be A, G, C, or T in DNA. So we have 4 amino acids, 4 nucleotides, so it can be here. So the probability is 4 different types multiply 4 multiply 4. So each time, each codon could be a combination of these 4 nucleotides. If you look at the codon, because the codon is shown using the RNA, So and in RNA we don't have thymine, so that's why you will see that here as A, C, U, and G. So again, when you are going to write these codons on the table, you will see the combinations such as this. And each of these codon is responsible for carrying one amino acid by the tRNA. Another thing which is important for you to know the reason that because we have 64 different codon, but we have only 20 amino acids, in that case we will have more codon than amino acids. Then what happens is that, for example, we have for phenylalanine we have 2 codon. This means that if the cell sees UUU, then the tRNA which is carrying the phenylalanine can come, or UUC. So in both cases, inside the cell, the gene, if there is a UUU or UUC, in both cases, the tRNA which is carrying the phenylalanine can come and attach. Or if it is a UUA or UUG, again, the tRNA which is carrying leucine can come and attach to the ribosomal protein complex, protein synthesis complex. If you look at this table, you will see some different color which has the meaning. The start codon is always starting with the tRNA carrying methionine, and the codon is AUG. So this is something that you should memorize. What is the start codon? AUG. And if you look at over here, we have 3 stop codon, URR, URG, UGR. So you can remember like that. URR, URG, UGR. This is the way that I remember. So any of these 3 different codon can inform the cell and the gene, the translation complex, that the tRNA is coming with a stop codon, which means that tRNA doesn't have specifically any amino acid. So the tRNA having the anticodon of UAA or UAG or UGA doesn't have any amino acids. Instead, it has a complex sending an information and showing that, hey, that's the time and we need to separate. And that's the end of the protein synthesis. Here we go. I suggest you go back and watch the video that we have already observed in the class regarding the DNA, regarding the protein translation. This is just a simple picture. As you see, the mRNA, the codon over here, and the anticodon, and the tRNA, and each time a tRNA having an amino acid will attach based on the complementary to the codon. So the anticodon will attach, and then the link between the 2 amino acids will form, and then the next, and then the next, and so on. So this tRNA comes to this binding site, and then the attachment of these 2 amino acids will happen, and then separation, and then the next one, and finally we will have the polypeptides formed at the end of the protein translation. And this slide explains what I already mentioned. And again, this slide. This is a practice of how to use a table of genetic code. Starting from here, you need to be able to predict or to say what is going to be translated for each codon. So you need to look at the table, for example, see there what is GCA in the DNA complementary to the mRNA. So that would be CGU. Be careful that we don't have thymine in mRNA. Instead, we have uracil. So the complementary strand for DNA in the mRNA for GCA is CGU, and then you look at the codon in the table for CGU and find out what is the CGU responsible in terms of amino acid, and then you will see that the CGU is carrying or is responsible to code arginine. Then you can translate by yourself just by using the table. There is a tutorial. You can pause the video, find the link using your PowerPoint slide, and study by yourself. Practice transcribing and translating using the Learn Genetics tutorial. So you need to be able to answer all these questions. Pause the video, think about the question, go back to your PowerPoint file, open the slide, and find out from the table that what is the codon that translates for aspartate. The second question, if mRNA contains the base sequence of this, then deduce the amino acid sequence of the polypeptide translated. Deduce the base sequence of DNA antisense strand from which the mRNA was transcribed. And if mRNA contains the base sequence of this, deduce the base sequence of the DNA sense strand. So all the questions related to make sure how you translate from DNA to mRNA or from mRNA to DNA. So you have to make sure you don't get confused or you don't make a mistake between thymine and uracil, between mRNA and DNA. Okay, here we go. Here are the answers. I'm not going to repeat the answers. You just need to pause the video before coming to this slide and write the answers, and then check your answers with this slide. The next question, transcribe this DNA strand into mRNA. I think this is so simple. Pause the video and answer the question. Yes, here we go. And the important is be careful about uracil once it is going to the mRNA. And another question, translate mRNA into polypeptide. So you know the DNA, you know the mRNA, then how do you want to translate mRNA into amino acids? So you need to look at the table and find, for example, what is the UGC translation. So you go here, U and G and C. So UGC from here. And for example, AAU, then you go AAU and AAU. So you just need to look at the codon and see what's going to be translated in the universal genetic code table. Okay, if you answered exactly the same way I answered, that means that you are completely wrong and you made the biggest mistake in answering these type of questions. Why? The reason is there is no translation from the mRNA to protein if there is no starting codon. So this means that first of all you have to find the starting codon, which is methionine. And what was the starting codon? AUG. Okay. Is there any other AUG over here? No, that's the AUG here. And another starting codon is here, AUG. So every time an AUG has the information regarding the translation of the protein. So if you start from here, then your translation would be from here. So you look at the AUG over here, that's the methionine, and the next would be CCU. So what is the CCU? CCU is here, proline, and then the next would be AAU, and the next would be GUC, and then the next would be AGG, GUU. And what is the termination codon? Here we go, UGA. You remember URR, UGR, UGA. So UGA is a termination codon. So if you want to answer this question, then you have to be careful about the start and finishing in the termination. So what would be the translation? Your protein started with methionine and then end up with the UGA as termination, and here is your sequence of the protein. That's it. This is the answer, starting with methionine and ending with UGA as the stop codon. So 6 amino acid in the polypeptide plus stop codon as here. So your polypeptide include only 6 amino acid: methionine, proline, asparagine, valine, arginine, and again valine.